lipid discussion a little bit, although I'm going to take a bit of a step back and talk about how we can make potential lipids. I think maybe what we're learning most of all is that we're not particularly creative picking Wikimedia slides. But the great news is I can skip all of my intro and focus on the detailed or mechanistic organic chemistry. I won't do that. Anyways, so lipids are important, membranes are important. When I'm talking about lipids and surfactants, we already had a really good explanation of this, but I tend to think about polar head groups and nonpolar fatty tails, which will partition preferentially to the air-water interface and will also form three-dimensional structures, as we've just seen. This has a lot of the same information that we just saw, but essentially, you've got your simple fatty acids that are used as your prebiotically relevant model systems, and then you can also make vesicles out of phospholipids, which are much more complex, but not particularly prebiotically relevant. A few other things about the vesicles that these two systems make. Fatty acids make vesicles, but they tend to be very pH and salt dependent and very sort of conditionally dependent, and also tend to be a little bit leakier than phospholipids do. Whereas phospholipid vesicles form at much lower concentrations and are stable under sort of a much wider range of conditions. But in the absence of the inclusions and things that modern life has, they tend to not allow they tend to be very impermeable. So one of the things that we're thinking about is, what if we had something that sort of explored the chemical space in between these two things? So you can think about phospholipids being robust but not very permeable, fatty acid vesicles being permeable but not very robust. But there's sort of this wide range of other possibilities that we're looking to explore. And in particular, obviously, we know that the head group of the phospholipids is very important, but also the fact that you can have single-tailed or double-tailed lipids is also important. So we're sort of looking for ways of making double-tailed lipids. And that's where prebiotic chemical synthesis comes into play, and looking at a simple abiotic synthesis of multi-tailed lipids. So when we're thinking about this, we have to pick environmental conditions. And as many people in this room know, our lab tends to like to use sunlight and water. And so the sun is a really good energy source because it is the largest energy source, even on a prebiotic Earth with the faint young sun and all of that. It's also a low entropy source, so you're not giving off a lot of, it tends to be very specific. And then in terms of environments, we like water and air-water interfaces in particular because they would have been widely available and relatively gentle. So just to talk a little bit about the young sun, many people know that when we're talking in the period of sort of prebiotic chemical evolution, when we're talking, the sun was quite a bit less luminous, about 25% less luminous than it is today. But there was still plenty of the UV radiation that is sort of high energy and useful UV radiation. And more of that high energy UV radiation, so not the super high energy stuff people were talking about before, but the sort of nice near UV energy, more of that would have reached the surface of Earth because we didn't have oxygen and because we didn't have the ozone layer shielding it. And so we've heard a lot about how that UV radiation can destroy things, but my point is it can also build things up. Yes. OK, and besides the fact that the sun was the largest energy source available, photochemistry is also fundamentally different from thermal chemistry. Thermal chemistry, you can think about just shaking molecules and cooking them to a point where they react. Whereas with photochemistry, it's very molecule specific, and you can excite one molecule, and the molecule next to it can be completely unaffected. So you don't have to heat stuff up, and you can get much sort of more high energy and out of equilibrium reactions to occur than you would get with thermal chemistry. And it's also molecule specific. So you can start with 3 molecules that look very similar. So you have hexanol, hexanoic acid, and 2-oxohexanoic acid. And you can see that only the 2-oxohexanoic acid with the extra carbonyl is absorbing light in the near UV region. And so it's very molecule specific. It's also incredibly environment specific. So these molecules, so pyruvic acid, which may be more familiar to biologists as pyruvate when it's deprotonated, is in the gas phase, absorbs light at about 350 nanometers. But as soon as you put it in water, it blue shifts slightly and absorbs at about 320 nanometers instead. So there's a shift that occurs. But you'll notice that as we add an alkyl tail, there really isn't much shift in the absorption cross-section. And so it's very much more dependent on the functional group than it is on the alkyl tail. And the photochemistry that these molecules follow, the mechanism is completely different in the aqueous phase than in the gas phase. And I'm happy to talk all of the details about mechanistic photochemistry if you want, but I'm only going to go into some of it. And so what we've been doing over the last few years is going through a whole set of these alpha keto acids, or oxoacids as I will call them, starting with the shortest, which is pyruvic acid, which just has this little methyl tail, and then a series of other molecules going up to the 12-carbon 2-oxo-dodecanoic acid. I'll be talking mostly about 2-oxo-octanoic acid just because you can purchase it from Sigma, and it doesn't have to be custom synthesized. So we have a very simple photochemical reactor. We use a xenon arc lamp, which is a good solar simulator, and we just shine light on a solution of our molecule in water. We can control the atmosphere, the gas composition of the reactor as well. But it's a very simple setup. But I like a simpler picture of what's going on. So you have, and here I'm just showing pyruvic acid because it's the easiest one. And so you've got pyruvic acid floating around in your solution. But one of the things to keep in mind is that pyruvic acid isn't just pyruvic acid in solution. You also get hydration reactions to form the geminal diol. And this geminal diol does not absorb light because you've removed the carbonyl chromophore. So you have to keep in mind that there's an equilibrium between the two of these. And it's dependent on the pH and the concentration of the molecules in solution, what the exact ratio is. But you still, unlike many dicarbonyls, you still retain a lot of your absorptive feature. And so you can still do lots of photochemistry with your oxoacids, where you wouldn't with an aldehyde or a ketone. And so your simple picture looks a little bit more like you've got pyruvic acid and then the diol floating around in solution. But if we hit that with sunlight, we can excite these molecules, and the excited state, so you excite to the singlet and you undergo intersystem crossing and internal conversion to the triplet state, for those of you who care. And then those molecules can react, the excited molecule can react with the other molecules that are floating around in solution. And so you can undergo a process called hydrogen abstraction, and you can do this from either the diol form or the carbonyl form. And essentially what you're doing is you're making organic radicals in solution. And there's a series of different radicals you can make. You can hydrogen abstract from the carboxyl group, or you can hydrogen abstract from this methyl group, which is something we've just shown can happen. But you get a series of organic radicals, and luckily we've already heard a lot about radicals. And essentially what you need to know is radicals are really reactive. They like to run into things and react. And here I'm showing the oxoacids with their alkyl tails back again. And it's worth noting that I'm demonstrating that the hydrogen abstraction has occurred at this beta CH2 group, but in theory it could occur anywhere along this alkyl chain. But just for ease of presentation, I'm showing it that way. And so if you have a solution where oxygen is present or other species, you can get quenching of these radicals or quenching of the triplet state, which will regenerate your initial oxoacid. So that tends to cut down on how much of the product formation you see. But luckily, prebiotically, we've established there isn't a lot of oxygen around. And so what you get instead is recombination to form oligomers dominating. And they are actually oligomers because I've got both dimers and trimers. And you get this especially in low oxygen environments. And so if we look at some actual data, this is just electrospray ionization mass spec for 2-oxo-octanoic acid. So before photolysis, we see the oxo-octanoic acid and its diol. And then after photolysis, the sort of major peak that we see showing up over here is this RR tartaric acid derivative, so a double-tailed lipid being formed. But you'll notice that that's not the only product we're forming. So we did quite a bit of mechanistic work to figure out what some of the other products we were making were. And so there's other possibilities that can happen. So instead of just getting these radicals, you can also have an intramolecular reaction that essentially kicks off the alkyl tail and you regenerate pyruvic acid from your longer-tailed oxo acid. And essentially, there are a couple of different processes that it can occur where you're generating multiple photoactive molecules in these oligomeric intermediates that are themselves photoactive. So you've started from a single, simple molecule in solution, but you're starting to generate all of these molecules that can go and do photochemistry and cross-react with each other as you go. And so like this is one of the very simple examples, but you can have the pyruvic acid that you generated via the Norrish type 2 reaction, and it can react with the oxo-octanoic acid. And you could get this guy, which is still your tartaric acid derivative, but this time you've got a 6-carbon tail and a methyl group. And so there are a series of reactions that can happen like that. And so this is as close to messy chemistry as a physical chemist is willing to get. But you end up with a series of different isomers with different molecules with 1 to 2 to 3 multi-tailed tails for your oligomers that are forming. And all of these, and I should say that all of these could be slightly different isomers, because with mass spec, you can't tell the difference between the two. Yeah, and so we've got this series of multi-tailed photochemical products, and it's sort of very easy to see this ready formation of multi-tailed lipids, even from a simple single-tailed precursor. And then, of course, we also have this tartaric acid derivative, which is the main product that we see in the mass spec. And then those of you who are keeping track at home, this is the 4th year I've put this slide up. But as we do this photochemistry, we also see self-assembly. And so we start with a clear solution that's below the aggregation concentration of the lipids. And as photolysis proceeds, the solution gets cloudy. And we're forming aggregates that are spherical in shape and monodisperse in size. And they're too big to be, so they're too big to sort of be what you would classically think of as a micelle. And so we have posited in the past that they are vesicles. We've been working to characterize those more. I think it's worth pointing out that even if they aren't vesicles, it's an interesting regime of self-assembly where you're getting these colloidal aggregates that are monodisperse in size and spherical and that last for years in solution. So we're sort of working on that. I can add that this happens even for your really short 2-oxohexanoic acid, which only has a 4-carbon tail. As you do photolysis and get these double-tailed or multi-tailed products, you see this aggregation occurring as well. And you see that for the mixture of oxo-octanoic and oxodecanoic acid. We don't see this for pyruvic for obvious reasons. It's just too short. And I'm working on getting some more information about this. And I should have, if I come back next year, this shouldn't be the same slide anymore, because we've got some good stuff coming. It's just not quite ready to share yet. And yeah, and so basically, we've been making, we're sort of exploring this path to making other molecules with sunlight and sort of a very ready synthesis of multi-tailed lipids from simple prebiotically relevant precursors. And so with that, I'd just like to acknowledge our group and all of the funding, and I'm happy to take any questions.
But I got a question on the way.
Okay.
Do you have any idea what sort of yields you're getting from this?
So we think we're getting, so it's a little hard to tell with, it's easier to tell with pyruvic because the NMR is much simpler. And so with pyruvic acid under anaerobic conditions for 5 hours, we get about 90% of the pyruvic acid is consumed. For the oxo acids itself, for the longer-tailed ones, it's probably closer to about 30%. But it's also harder to tell, so the error bars on that yield are, yeah, and that we can really only monitor the decrease in the starting material. So I don't know the ratio other than relative mass spec of the products we're making. So, yeah.
So the first question is related to our PWR proposal. So if you have ever experienced any sort of UV effect on vesicles. So if you know if something happens on vesicles already formed when you shine light on them. And then I have another question. So we have seen that phospholipids have two tails. So I was wondering, do you think that, do you envision a phosphorylation reaction happening after the formation of a double-tailed fatty acid? Or would you think that first a fatty acid is phosphorylated and then it forms a second tail?
Yeah, so with the sort of first question, I haven't done any work sort of on vesicles and doing photochemistry on sort of preformed vesicles, although it would be interesting, and I have some results that I didn't talk about, where you can also get the cross reaction with the photoactive oxoacid and a fatty acid, and you can get that cross product as well. So I don't really have an answer to your first question because I haven't really done anything with it. As to the second one, let's see. What was it again? Sorry. I had an answer, then it disappeared.
Yeah, if you think that the phosphorylation reaction happens.
Oh, right. So I think it would be hard to phosphorylate the starting material of the oxoacids because it is so molecule specific. You're going to change the electronic structure, and so the mechanistic, the excitation and then the photochemistry that happens may not be the same. Bradley would know more about phosphorylating the products. I think even if you could phosphorylate the products, as we were talking about earlier, the issue is going to be adding the choline group, I think, because I don't really know how that would happen. Yeah.
Hi, I'm just curious about the whether there is any published or unpublished prebiotically available pathways for the alpha keto acids that you proposed.
Yeah.
That's my first question. And the second question is when you talk about the alpha keto acids forming these vesicle-like structure, do you know if they're multilamellar or unilamellar type of vesicles? Thanks.
Yeah. Okay, so the first part, so pyruvic acid is a very prebiotically relevant molecule, so the shortest one. And you can make it in hydrothermal vents, you can make it a bunch of different ways. And it's been seen in meteorites and things like that. I believe other oxoacids have also been seen in meteorites, perhaps not these ones with the alkyl tails, but they are also the right length for sort of a Fischer-Tropsch type synthesis as well. So I can't say 100% that these specific oxoacids have been seen prebiotically, but they're certainly prebiotically reasonable. And then the second question was, just talking about whether we know if they're lamellar or not. We haven't been able to characterize, we've been trying for a long time to get access to a cryo-EM to see the lamellar structure, so I don't have any information for you about that. Yeah, I don't know.
I want to ask about time of exposure in the photochemical reaction.
Yeah.
For how many seconds or minutes or hours you expose these biomolecules for photoactivation? Because in evolutionary history, Earth was our biomolecules were exposed for that kind of physical things for a long period. So what is the probable effect when we expose biomolecules for that kind of physical parameters?
Yeah, so these particular experiments are 5 hours long in the 450 watt xenon arc lamp. That's picked mainly because I can do that reaction in a day, because it just works out better. The interesting thing to note is that because stability of your products is always going to be a question, so these products here with the oxoacids absorb light, but you'll notice that oxoacid functionality is not in the products, so they don't have chromophores, so they're not going to absorb light in that near UV region, so they ought to be relatively stable and UV protected. I mean, not from really high intensity UV light, but at least from sort of near UV light. And then
Nope, that's it. Thank you very much.